molecular cloning stands as a cornerstone technique in the vast field of genetic engineering. This revolutionary process enables scientists to isolate and amplify specific DNA sequences, essentially creating identical copies of DNA fragments. The implications of molecular cloning are far-reaching, spanning diverse scientific disciplines, from medicine and agriculture to environmental science and beyond. At its core, molecular cloning empowers researchers to study genes and their products in exquisite detail, unraveling the complexities of gene function and regulation. This knowledge translates into groundbreaking applications, including the development of novel diagnostics and therapeutics, the engineering of crops with enhanced yield and nutritional value, and the production of valuable biomolecules like insulin and growth hormone. The impact of molecular cloning on our understanding of life's blueprint is undeniable. It has revolutionized biological research and paved the way for countless scientific advancements. As we delve deeper into the intricacies of the genome, the importance of molecular cloning will only continue to grow, further shaping the landscape of science and medicine for generations to come. Traditional cloning, often referred to as restriction enzyme-based cloning, represents the bedrock of molecular cloning techniques. This time-tested method involves a series of well-defined steps, each crucial for the successful insertion of a desired DNA fragment into a vector. The process begins with the digestion of both the target DNA fragment and the vector with specific restriction enzymes. These enzymes act like molecular scissors, recognizing and cleaving DNA at specific sequences, generating compatible ends. The digested DNA fragments are then joined together by DNA ligase, forming a recombinant DNA molecule. This recombinant DNA molecule is then introduced into a host cell, typically bacteria, through a process called transformation. Within the host cell, the recombinant DNA molecule replicates, creating numerous identical copies of the target DNA fragment. Traditional cloning, while effective, can be laborious and time-consuming. The reliance on restriction enzyme sites can also limit the flexibility of this method. However, its reliability and versatility have solidified its place as a fundamental tool in the molecular biologist's arsenal. Gibson assembly emerges as a powerful alternative to traditional cloning, offering a more streamlined approach to assembling DNA fragments. This method bypasses the need for restriction enzymes and ligation, relying instead on the remarkable properties of three key enzymes: T5 exonuclease, fusion DNA polymerase, and Taq DNA ligase. The process begins with designing DNA fragments with overlapping ends, typically 15 to 40 base pairs in length. These overlapping regions are complementary to the adjacent fragments, facilitating their assembly. The DNA fragments, along with the three enzymes, are then combined in a single reaction mixture. T5 exonuclease initiates the assembly process by chewing back the 5' ends of the DNA fragments, exposing single-stranded regions. These single-stranded regions, guided by their complementary overlaps, anneal to their respective partners. Fusion DNA polymerase then fills in the gaps, while Taq DNA ligase seals the nicks, creating a seamless, fully assembled DNA molecule. Gibson assembly offers several advantages over traditional cloning, including its simplicity, speed, and ability to assemble multiple DNA fragments simultaneously. Its efficiency and versatility make it an attractive option for various molecular cloning applications, particularly those involving large DNA constructs. Section 4. Gateway cloning versatility and efficiency. Gateway cloning, developed by Invitrogen, now Thermo Fisher Scientific, presents a highly efficient and versatile cloning system based on the site-specific recombination properties of bacteriophage lambda. This method relies on two key reactions, the BP reaction and the LR reaction. The BP reaction serves to clone a gene of interest into a specialized vector called a gateway entry vector. This reaction utilizes the BP clonus enzyme mix, which mediates the recombination between ATB sites flanking the gene of interest and ATP sites present in the entry vector. The resulting entry clone harbors the gene of interest flanked by ATTL sites. The LR reaction, facilitated by the LR clonase enzyme mix, facilitates the transfer of the gene of interest from the entry clone to a destination vector. This reaction exploits the recombination between ATL sites flanking the gene of interest and ATR sites present in the destination vector. The final product is an expression clone, ready for downstream applications. Gateway cloning offers numerous advantages, including its high efficiency, directional cloning, and the availability of a vast array of destination vectors tailored for various applications. This system has gained immense popularity for its versatility and ease of use, simplifying complex cloning strategies and accelerating research progress. Section 5. Topo cloning, rapid and straightforward cloning. Topo cloning provides a rapid and straightforward method for cloning PCR products directly into a linearized vector. This method capitalizes on the unique property of ToC-DNA polymerase, which adds a single deoxyadenosine or A overhang to the 3' end of PCR products. Topo vectors are linearized vectors containing a single 3' deoxythymidine or T overhang complementary to the A overhang of PCR products. These vectors are precharged with topoisomerase I, an enzyme that catalyzes the ligation of DNA fragments with compatible ends. The cloning process is remarkably simple. The PCR product is mixed with the topo vector and incubated for a short period. Topoisomerase I mediates the rapid ligation of the PCR product into the vector, completing the cloning process. Topo cloning offers several advantages, including its speed, simplicity, and high efficiency. Its reliance on PCR products makes it particularly well suited for cloning genes directly from cDNA libraries or genomic DNA. Section 6. Conclusion. The future of genetic manipulation. Molecular cloning techniques have revolutionized biological research, enabling scientists to manipulate DNA with unprecedented precision and efficiency. From the traditional restriction enzyme-based methods to the more recent innovations like Gibson assembly, gateway cloning, and topo cloning, each approach offers unique advantages and caters to specific experimental needs. As our understanding of molecular biology continues to expand, so too will the development of novel cloning strategies. These advancements will undoubtedly enhance the speed, efficiency, and versatility of molecular cloning, further empowering scientists to unravel the complexities of life and develop groundbreaking applications in various fields. The future of genetic manipulation is brimming with possibilities. As we continue to push the boundaries of scientific exploration, molecular cloning techniques will remain indispensable tools, shaping the landscape of scientific discovery and driving innovation for generations to come.